devices, the resilience is needed to face global challenges. A part of COVID-19, Indonesia still has many unresolved infectious disease problem. Several infectious diseases such as tuberculosis, malaria, dengue, HIV, AIDS, hepatitis, and etc. Still require innovation in reducing their incidence. This is where our researchers are. On this occasion, we are lucky to invite expert researchers from Shizuoka University to share their knowledge. And at this mini symposium, our researchers from the Research Center for Vaccine and Drug of BRIN were also present to become plenary speakers. I hope this morning moment can bring up new ideas and solutions against infectious diseases. I also hope that the collaboration that has been built with the Shizuoka University can continue and can motivate all of us to continue to be enthusiastic about collaborating. I think that's enough of my opening. Enjoy the webinar. Thank you. Wassalamualaikum warahmatullahi wabarakatuh. Om Santi Santi Om.
Waalaikumsalam warahmatullahi wabarakatuh. Immensely appreciated, Prof. Indi. So let's move on to our head of Research Center Vaccine and Drug, and also actually host department for this event. We would like to invite Dr. Masteria Yunifilsa to have some speech to welcome our guests this morning.
Thank you, Dr. Rika. Good morning, ladies and gentlemen. First of all, I would like to say my apologies to Dr. Enoch Park and all of the head of research center in Research Organization for Health. I cannot attend by offline this morning in the symposium because I missed my train last night. Then I just arrived from Jogja to Jakarta this morning. So, but directly 1 hour I will be arrive in Laptiap. So I would love to express my gratitude to Prof. Dr. Niluh Putu Indi Darmayanti as the chairman of Research Organization for Health for joining us today, and our guest, our honored guest today, Prof. Enoch Park, together with his team who came all the way from Japan, from Shizuoka to Indonesia and visit our research center and discuss the collaborations about the infectious disease with our research center. And thank you also for all the participants who joined us today. Thank you for coming. So the word change promptly, we are benefited apparently as a change leverage of our quality of life. However, we do not ignore that any kind of elevation takes consequence, one of which is involvement in every aspect, and that is obvious in research. As such as collaborations stop the options. So commemoration of National Technology Awakening Day that we called it Hakteknas, which annually falls on August 2nd, remind us the urgency of building a strong basis of joint research and harness it as steps to reach higher levels. So we understand and accept our imperfections, but we acknowledge and realize more we have got a lot to contribute. What Indonesia is yet to fulfill in aspect where our collaborators stand strongly. So what in other hands, we will compensate by contributing our best together to reach much better goals that are impossible to hold alone. Frankly, I do not expecting this meeting, which was set as informal, become so big with many parties involved. But I am beyond happy to say that it's exemplary work and need to see many more of scientific collaborations and meeting in near future, and so our regular event. Thank you for all participants involved today. This happens as everyone participates enthusiastically we will definitely learn more insight from this mini symposium, biotechnology applications and against infectious disease. May our session run successfully and enjoy the mini symposium today. Thank you very much, Dr. Rika.
Yeah. Thank you, Dr. Masteria Filsa. And I think this is the most awaited sessions that we are having Professor Enoch Y. Park here. But before he delivers his presentations, which will bring a theme of sustainable fight against infection disease causing viruses, I would like to share with all the participants how he has been so far, which have a very outstanding resume in my hand. He's now served as a director of Research Institute of Green Science and Technology, Shizuoka University, and has been served for a long time as researcher and the Central in Miwon Corporation Limited, working on the methane gas productions, and also after that becomes associate professor, visiting professor, and so on. And his research expertise are covering gene expression, virus-like particles, silkworm biotechnology, but we would like to highlight in this morning, bioprocess, nanobiotechnology, and virus detections. And of course, he's a valued member of several academic societies. And about the counselors, he became the member of the Society of Bioscience, Biotechnology, and Biochemistry Japan. And this make me wonder as well that he's excellent in three languages, Korean, Japanese, of course, and English. He's got so many awards, and you don't ask me about the publication because it's very huge. Without much further ado, you are most awaited, Professor. Please, you can, floor is yours. You can start to present.
Thank you very much for your kind introduction. Good morning. It's okay. And it's my great pleasure to meet Professor Indy and Director Masteria, and also all participants in this meeting room. And even if it is offline, however, it's my really a great pleasure, because after COVID-19, I first go out from Japan to overseas, this first trip. Thank you very much. Also, I congratulations for an extensive reform of BPPT and reborn as a BRIN. I really think you have a great endeavor for this reform. Okay, I hope this BRIN will prosper more and more and become one of the greatest institutes in the all over the world. I would like to introduce a short history, BPPT and our research institute, Green Science Technology. We have reached the first 2014 March. At that time, director was Dr. Marjan. And 2014 April, Professor Marjan and Professor Marjan and Dr. Enya, Dr. Ungul, Dr. Soni will visit our university and discuss about extensively about how to promote international collaboration. And same year, September 16, we have joined MOU in BPPT in Jakarta office. And after then, we discuss every year. 2015 August, I visited BPPT again. 2016 also, our staff, almost I think 10 people will visited the BPPT and discuss and held a symposium. However, 2020 February is the last time because 2020 March, all country banned foreign movement. Therefore, this is the first time after 2.5 years later, I visit again. It's very, for me, it's very a great thing for me to meet you again. And also one more thing is we have a alumni in Indonesian student. We have Dr. Carl, he's now working in Kobe University. Dr. Liz is working in University of Al-Azhar, Indonesia. And Dr. Johnny is working in BRIN also. And Dr. Spame is working in Kazamada University. Dr. Todi, today he will talk some, his research. And Dr. Indra is not doctor right now. However, 1 month later, he should be doctor. Therefore, he joined and for virus detection. Dr. Rizal also joined for virus detection in this symposium. And my talk today is 3 points. One is the detection of mosquito-borne viral disease. And the second talk is Silkworm Biofactory. Third is a Sprout project. The first, this virus detection is for international collaboration work between our lab and Dr. Sabah, supporting by a next joint international research. We think about why virus cannot detect like this pH meter. This pH is a very small one, however, very tip here. They can sense pH and the digit here very easily. Why pH virus cannot detect like this pH meter is our strong question. In several years, we have developed a new detection system. This is electrode, the molecular recognition element. This electrode was deposited several kind of nanomaterial, at least 2 or 3 layers here on the surface of this electrode. In the absence of virus, the impedance is very low. However, in the surface, the presence of target virus on the surface binding here, impedance jump up. This is the main principle of this detection research. Therefore, we use this kind of signal transduction for the virus detection using this kind of electrode. Therefore, detail of this research will be talked by Dr. Rizal on the Mr. Indra's a similar research achievement today. He will deliver talk today. Okay, I move to second talk. Silkworm Biofactory is a silkworm. They now you cannot display this. So they cannot display this movie. Oh. Oh, cannot. Oh, yeah. Okay. Stop. It's okay. Oh, okay. Okay. This is now silkworm. They enjoy their dinner. This kind of material is for silkworm artificial diet. They use they like this kind of food, and the length is almost a finger size, and almost 5 centimeter, and thickness almost 8 millimeter. A very good one. And why we interested in silkworm? This is a very simple story. Protein productivity is defined as a specific productivity times cell concentration. A specific productivity, it means 1 cell, 1 hour, how much protein was expressed. It's a specific productivity. Therefore, if you have 1 cell in reactor, we can get 1 specific productivity, 1 QP. However, we increase 20 times cell concentration, we increase 20 times specific productivity. However, we use silkworm. Silkworm is very small, almost a finger-sized insect. And the inside of this silkworm, as you see, more than 1 million cells is living here. Therefore, in the term of protein productivity, silkworm is the best way to express protein with very high yield and the mass production. This is why we focus silkworm. Forever, to use this kind of silkworm, we have to overcome a very big problem, because to introduce foreign gene into silkworm is very difficult, very tedious. It takes a long time. Therefore, we develop new method, Bombyx mori nuclear polyhedrovirus vacuum system. This vacuum is shuttle vector between E. coli and silkworm cell or silkworm itself. Therefore, we manipulate the gene in E. coli level. Therefore, very easy, very simple, and very fast we can get the gene. And after we get the backmid from E. coli, we extract and directly inject the silkworm. Almost 4 or 5 days later, we can observe like this full expression of, in this case, a green fluorescent probe, GFP protein. We can observe this green color, green fluorescence here. Also, same pupa also almost 100 hours later, we can express full expression of GFP here. And this is the hemolymph of silkworm. It's contained full of GFP also here. And this kind of research was the first achievement in our lab in the world. Until now, very difficult to introduce foreign gene to silkworm. Using this kind of new vacuum system, we can express many genes in silkworm very easily, shown here. As a security protein shown here, we usually focus on eukaryotic gene. Here, human type, rat, human. Is all animal or human mammalian cell origin. Therefore, every protein was expressed very easily and purified, and we get the biological function, very high activity. Also, as membrane protein shown here, human origin, protein receptor, also immune cell surface receptor, also GPCR is very famous. Also, we confirmed the expression and purified and confirmed the biological function. It's a good function. And also, we are focusing now virus-like particle. As envelope virus-like particle, Rouse sarcoma, we have many research achievements here. Also, influenza virus like particle, also we get displayed hemagglutinin and neuraminidase on the surface of this VLP. In dengue virus like particle, also Dr. Dodi is expert in this field. As a non-enveloped VLP shown here, HPV, a human papillomavirus VLP, it already is commercialized, and all of the people all the world, many people use this as vaccine already. The virus-like particle also expressed a very high yield in silkworm. The canine virus also we expressed in silkworm recently. Yeah, why we focus on viral-like particle? Shown here, the VLP is composed of a viral protein and capsid protein. The inside of this VLP is empty. There is no genetic material inside of the VLP. However, virus, they have a genetic material inside of the VLP virus. Therefore, they can infect. However, VLP cannot infect. However, VLP can induce immune system. Therefore, if you have a very good target for vaccine candidate. Therefore, all over the world try to make VLP as vaccine candidate now. This is one example. In our laboratory, we published that dengue VLP like this, almost 35 or 40 nanometer size, and the surface of the VLP is very smooth. However, norovirus-like particle almost is 50 nanometers, 40 nanometer. On the surface of this VLP, as you confirm here, the rough, not smooth, because this is a non-enveloped VLP. Canine VLP also, the surface is very rough, because only protein, they don't have any envelope. Therefore, silkworm can produce this kind of VLP very easily and purify, very good purity. We also confirmed. And now we are challenging how to many antigen can display on the surface of VLP, a platform. And as you know, in dengue also, we have the 4 cell types. Therefore, we need the 4 kind of antigen should be displayed on the surface of this VLP. How to display different kind of antigen and can display on the surface? We are now thinking about a spike catcher, spike tag system. A spike catcher, they easily, in room temperature, they easily bind together by isopeptide bond. Therefore, in spike catcher, we fuse protein A. In this case, we use a VLP gene. And the spike tag here, we fuse the protein B. In this case, we use antigen gene. Therefore, 2 different kinds of construct we prepare. And we inject this backmid to a silkworm. In this case, in here, Venus is kind of a GFP shown here. We fuse Venus to spike catcher and express shown here. Actually, we cannot observe red color of Venus color. Here, green color we can observe. It can express. And M cherry was fused with spike tag here. M cherry is red color. Therefore, M cherry, the red color we can observe. However, we cannot find Venus fluorescence. However, we co-infected these 2 kinds of backmid, one silkworm, we can observe red and green. Therefore, this 2 kind of protein was expressed in one silkworm. Also in here, as is shown here, here, this one and this one express a Venus, and this middle one is just expressed M cherry. And this M cherry and the Venus, 2 backmid were co-infected in one silkworm shown here. Color is different, not green, not red, kind of yellow color. Therefore, it 2 color, 2 gene, 2 protein was fused in a isopeptide bond. Therefore, they formed one VLP, and around and the surface of the VLP, the spike tag was bonded binding here. Therefore, M cherry was displayed on surface of this VLP. We can conclude. Therefore, we can conclude VLP can be displayed using this kind of spike catcher spike tag system. And this is recently we get the research achievement. We identify a new vaccine candidate against the white spot syndrome virus. This WSS4 is very severe damage in Japan, especially in kuruma shrimp. And very usually 2 or 3 days, all shrimp was dead by this virus. Therefore, we screened many kind of antigen against this WSSV and find a good candidate. And the candidate vaccine was expressed in silkworm pupa, and pupa was powdered and part of the pupa was mixed with shrimp feed. And the shrimp feed were orally administered to shrimp. And then the result, this red color, in pupa, a relative survival rate, RSR, parameter is almost more than 81%. In this case, 100% of survival. Therefore, it's very good result. Therefore, this kind of thing, if you express antigen on pupa, and pupa was orally administered animal, it can good effect. Therefore, and this result can prove defense against WSV. I think I see fully practically use this kind of vaccine for against shrimp. Therefore, this silkworm is, I think we think silkworm is a good biofactory, because silkworm we know well produce silk product. In Japan, we have many idle land. Let's plant mulberry tree, and we can get silkworm from noble silk product. Also, we can get recombinant protein, and we can utilize the pupa, and we can get a mulberry tree is a supplement for healthcare. Usually in mulberry tree, fruit is very famous for antioxidant. This kind of thing was how we can more expand it to our local society. For example, some area we can express pharmaceutical product. Some area we can express food enzyme. Some area we can express an animal vaccine. Therefore, we can revitalize local region using the silk biofactory. It's a first industry, and this kind of product was used for pharmaceutical industry or chemical industry as a secondary industry. And also, this kind of product was used for a commercialization. It's kind of a creation of a bioproduct by industry. Therefore, we call it tertiary industry. Therefore, we can think this biofactory, this biofactory can create a sustainable bioindustry, namely the 6th industry. Also, we can contribute SDGs 2, 3, 9, and 12. All these systems are all sustainable. Silkworm, you can reuse. You can get egg and hatched again, and we can use silkworm, and also we prepare egg with sustainable. Also mulberry leaf. Also we can harvest the mulberry leaf in Indonesia 365 days. Every day we can harvest the leaves. Therefore, anytime we can rear silkworm. Therefore, I'm suggesting how about to apply this science and technology research partnership for a sustainable development international collaborative research program. We call the SATRAPS. And I think a good partner with you, BRIN and our laboratory. Because we are working a long time, we collaborate about a VLP or a silkworm biofactory system. Therefore, I'm strongly suggest to collaborate this kind of research with you. This is my summary. Silkworm platform has great potential targeting for a sustainable fight against the infectious virus causing disease. And also, I like this word, silkworm related to biolot from silkworm. So it is our university picture, and thank you for your very kind attention. Thank you very much.
It was super, professors, and we are not collecting questions for this session. However, I suppose that you have written down his email address, so feel free to contact him directly or after these sessions. And what I'm going to highlight before we move on to next session is from this speech only, we realized not only are we posted with the reasons of advancement of building biofactory with so bright ideas, but we came into realization that some living reservoir, especially those very near to us, both digging up scientifically and that might be commercially or economically gain for us. Thank you so much. And next is the photo session, right? Photo sessions. Participants who are attending, please unmask yourself if you are wearing one, because that's not, yeah. This is things to remember who are going to click the pictures, the committee. How many layers are we? 4, 5, or 1 layer? On my count, 3. On my count on 3. 1, 2, 3. All good? Thank you so much for all participants. We now move on to the discussion sessions, which will be led by moderators for first sessions. Our moderators is Dr. Sabar Pambudi, where he is acting as our chair for vaccine research group. So Dr. Sabar, floor is yours.
Thank you, Dr. Ika. Ladies and gentlemen, it will be for the next session today for invited speaker. So before I start, I would like to introduce our speakers. So this morning, one invited speakers from Shizuoka University will deliver the one presentation. His name is Dr. Takatsugu Miyazaki. He graduated from Tokyo University of Agriculture and Technology from for his bachelor, master, and PhD degree. After he graduated from TUAT, he got some position from Japan Society of Promotion Science, and he continued as assistant professor in Research Institute of Green Science and Technology, Shizuoka University, from 2016 until now. So they have a lot of publication, and today he will share about one topic. The title of the presentation is Structures and Function of Novel Glucosidates Discovered from Bacterial Genomes. So I would like to invite him to give the lecture. So Dr. Miyazaki, the time is yours.
Thank you for a kind introduction. Good morning, everyone. I am Takatsugu Miyazaki, an assistant professor of the Molecular and Biological Function Research Core, or RIGST, Shizuoka University. First, I would like to thank the committee for giving me the opportunity of my presentation. So, as introduced, I obtained a PhD at TUAT and joined RIGST at the Shizuoka University 6 years ago. I am interested in carbohydrate active enzymes like glycosidases and glycosyltransferases and their 3D structure. So I have been studied protein glycosylation and glycoengineering with Professor Park, and microbial and insect glycoside hydrolases using enzymological techniques and protein X-ray crystallography. Recently, I started to use cryo-EM for protein structure analysis. I now belong to Molecular Biological Function Research Core at RIGUST. Our research core aims to contribute to the achievement of SDGs number 3 and 9 by technology development for the application of biological and molecular functions to improve health and extend healthy life expectancy. My team analyzed three-dimensional structure of proteins of interest. Well, today I would like to introduce my recent studies on two novel glycoside hydrolases. One is alpha-1,2-glucosidase, and another one is alpha-1,3-glucosidase. First of all topics, I will explain the research backgrounds and purpose. Carbohydrates are involved in various biological functions. Energy storage materials such as starch and glycogen, osmolytes, cell walls, and shell components like cellulose and chitin. And intercellular and intermolecular communications involve protein N and O glycosylation in mainly eukaryotes. They are also related to pathogenicity, diseases, and cancers. Moreover, some carbohydrates, especially oligosaccharides, act as prebiotics that induce the growth of beneficial intestinal microorganisms. Glycoside hydrolases, GHs, are enzymes that catalyze hydrolysis of glycosidic linkage in glycoside, oligosaccharides, and polysaccharides. They are involved in degradation, synthesis, and reconstruction of carbohydrates. For example, starch is a glucose polymer. It is hydrolyzed by alpha amylase and produce malto-oligosaccharides, also called dextrin. These oligosaccharides are further hydrolyzed by alpha-glucosidase to produce monosaccharide glucose in our bodies. Some GHs have transglycosylation activity and form oligosaccharides. So they can be applied to synthesis of various oligosaccharides. There have been reported more than 200 GH activities to date. GHs are classified into GH1 to GH173 families based on their amino acid sequence homology and summarized in the KZ database. Each family contains GHs with different activities and substrate specificities derived from various organisms. Some families contain viral GHs. For example, GH31 contains alpha-glucosidase, alpha-xylosidase, and alpha-galactosidase. Each GH family contains a tremendous number of putative enzymes encoded in the genomes of various organisms that had not been characterized yet. So we are aiming to discover GHs with novel activities from mainly microorganism genomes and to elucidate their structure-function relationships. Okay, next I would like to present the first study, structure and function of GH65 alpha-1,2-glucosidase. GH65 is a special family which contains not only GHs, but glycoside phosphorylases, GPs. This enzyme are active on alpha-glucosidic linkages. GHs cleaved glycosidic bond using a water molecule. In contrast, GPs cleaved glycosidic bond using inorganic phosphate and produce glucose 1-phosphate. GPs catalyze not only phosphorylases, but also reverse phosphorylases. So they can be applied to synthesize various oligosaccharides. Therefore, we became interested in GH65 family and searched for a new enzyme. This is the phylogenetic tree of GH65 enzymes. GH65 is divided into the bacterial GPs and eukaryotic GHs. All bacterial enzymes are GPs acting on mainly glucose disaccharides such as maltose, trehalose, cogibiose. And all eukaryotic enzymes are GHs, like trehalase. We found a protein gene which has low sequence identity with both bacterial GPs and eukaryotic GHs in the genome of soil bacterium Flavobacterium johnsoniae. So this was targeted. We cloned the gene from this bacteria and expressed in E. coli as a recombinant protein and purified, and then investigated its activity for glucose disaccharides, described here. We analyzed using thin layer chromatography, unacceptably, this enzyme degraded only alpha-glucobiose, cogibiose, and produced only glucose, but not glucose 1-phosphate, unlike other non-bacterial GPs. So this enzyme is not GP, but GH. Next, we performed X-ray crystallography of this enzyme to elucidate structure-activity relationships. We successfully determined the crystal structure of alpha-1 glucosidase. This enzyme was found to form a hexamer. This is a monomer of this enzyme. And as you can see, the whole structure resembles non-bacterial GH65 GPs, cogibiose phosphorylase and maltose phosphorylase. We also analyzed the structure of this enzyme complex with glucose product, glucose in its active site. Active site is the center of this domain. This enzyme has 2 glutamate residues in the active site. They are thought to be essential for hydrolytic reaction. And we identify the residues which are involved substrate binding. So we relieved how this enzyme recognizes and hydrolyzes cogibiose in the atomic level. Next, we would like to introduce the second study, structure function of alpha-1, 31 alpha-1 3-glucosidase. GH31 family comprised of enzymes active on alpha-glucosidic linkage, such as alpha-glucosidase, like intestinal maltase, and xylosidases. This is the phylogenetic tree of GH31 proteins. Similar to the previous study, we targeted the protein that belonged to function unknown protein cluster. and have low sequence identity with reported GH31 enzymes. We clone a gene from Lactococcus lactis. This is lactic acid bacteria, and expressed in E. coli, and then investigated its activity for glucose disaccharides and sucrose. This enzyme hydrolyzed alpha-1,3 glucobiose nigerose most efficiently, and also hydrolyze cogibiose and maltose with lower efficiency, although most of known GH31 alpha glucosidases have the highest activity towards alpha-1,4 linkage of maltose. We also determined the crystal structure of this enzyme using X-ray crystallography. This enzyme also forms a hexamer, and its monomer resembles the known GH31 enzymes, including human alpha-glucosidase, human intestinal maltase, and human lysosomal glucosidase. In addition to the crystal structure, we had the opportunity to analyze the sample using cryo-EM. This is a cryo-EM machine. We successfully observed the hexameric structure of this enzyme at 2.73 angstrom resolution. This is a relatively high resolution. And the whole structure was almost identical to the crystal structure. We also analyzed the structure of an inactive mutant, D394A, of this enzyme, complexed with substrate nitroligosaccharides, also determined by X-ray crystallography. This figure shows nitrotriose bound structure. Nitrotriose was observed at the active center here. We identify the residues which bind nitroligosaccharides substrate. Nitroligosaccharides is green one. This enzyme strictly recognizes nitroligosaccharide by hydrogen bonds and hydrophobic interaction. Interestingly, tyrosine 99 here, this is located in the neighboring protomer in the hexamer. This residue is involved in substrate recognition. So the hexamerization is important for its enzyme activity, enzyme specificity. Okay, this slide shows the conclusions. By the phylogenetic analysis of the GH families and mining bacterial genomes, 2 novel bacterial alpha-glucosidases were discovered, and we revealed the molecular basis for the substrate specificity and the reaction. About GH65 alpha-1,2 glucosidase, this is the first bacterial GH belonged to this family, and we reported the first 3-dimensional structure of GH belonging to GH65 family. Because there had been no report on enzymes specific for cogibiose, a new EC number was assigned to this enzyme. Next, GH31 alpha-1,3 glucosidase. Its specificity for alpha-1,3 glucosidic linkage is higher than any other alpha-glucosidase from prokaryotes and eukaryotes. This is also first 3D structure of bacterial alpha-1,3 glucosidase. Alpha-1,2 glucosides and alpha-1,3 glucosides are rare in nature compared to alpha-1 for glucosides like maltose and starch. And these components found in polysaccharides produced by some bacteria and fungi. So we think that these enzymes maybe contribute to degradation of such polysaccharides. We will continue to search for GHs with novel activity and elucidate their structure-function relationship. And we aim to apply these enzymes to synthesis and processing of useful carbohydrates. These researches were supported by nice collaborators. Thank you for attention.
Thank you, Dr. Miyazaki, for the nice presentation. It's very great to see your results during your activity in Shizuoka University. So we have several minutes before we go to the next session. If there is some question from the audience, we have like maybe 3 minutes. If not, maybe I have one question to Dr. Miyazaki. So as you mentioned that GH mainly have some activity to catalyze or hydrolyze the glycosidic bond in complex sugar. And we know that the function of this enzyme is including degradation of biomass such as cellulose or starch. And I found also there is another function of this enzyme regarding in antibacterial defense strategy or even in pathogenesis of in some viruses. So in your future study, is there any some other approach to see the function of this enzyme regarding for like antiviral activity by using this enzyme? Is there any possibility to use this enzyme for this kind of activity in the future?
I think when I have a started on some polysaccharide derived from pathogenic bacteria, we can degrade these polysaccharides using specific GHs in the future.
Okay. Yeah. I think that's a very interesting point. We can use this kind of enzyme not only for industry but also for health activity. I think also that's possible. I think maybe in the future. So if there is no other question from the audience, I will close this session from Miyazaki. So please give applause to him. Thank you very much. To him, thank you very much.
Terima kasih.
Thank you. Arigatou gozaimasu. So next to the session, go back to Dr.
It was so lovely presentations from Dr. Miyazaki. Next to the presentation sessions, this time will be led by Dr. Tarwadi, where he himself is the lead of research center of adjuvant therapy. So now floors and times yours, Dr. Tarwadi. Please go ahead.
Check. Is it okay? Yeah. Okay. So let's start again. So, ladies and gentlemen, we have a second session, and we have approximately 1 hour for 3 speakers. So hopefully every speaker will spend about 15 minutes. And the first speaker is Dr. Engineering Saikhul El-Muttaqin. He belongs to the Research Center for Vaccine and Drug. And right now, he's doing postdoctoral in Shizuoka University. He will present polyaniline-based electrochemical impedance spectroscopy, biosensors for virus detection. So, Dr. Isa, please, the time is used roughly 15 minutes.
Selamat pagi, Assalamualaikum warahmatullahi wabarakatuh. Thank you for everyone. Thank you, Pak Tarwadi, for the introduction. Yeah, today I would like to presenting about my research entitled Polyaniline-Based Electrochemical Impedance Spectroscopy, AIS, Biosensor for Virus Detection. My name is Rizal. I'm currently doing postdoctoral period in Park Sensei Laboratory, Shizuoka University. So this is the very short introduction about as you know, the infectious disease as the main cause of significant increase in pathogenesis and death throughout the world. And we're just suffering from COVID-19 right now. And the golden standard for virus detection is like a viral cultivation, ELISA, and also what we right now know better, polymerase chain reaction or RT-PCR. But as we know that those gold standard method still suffering from some disadvantage, like a strict laboratory condition requiring well-trained staff and expensive instrument and also time-consuming process. Therefore, biosensor offers rapid, accurate, dependable, and miniature device for virus detection that probably will improve the capacity test while in the future we will have another pandemic. We hope not. We hope not, of course. So this is the kind of the simple overview about the electrochemical biosensor. Since the beginning of introduction of the glucose biosensor, actually the biosensor for infectious disease become more famous. And as you can see here, the illustration is almost similar what we already know about the antigen COVID-19 swab test. It's basically it's kind of the decoration of the electrode here. And we have electrode, of course, because it's electrochemical sensor. We need electrode that conductive that could deliver the electron transport. Here, it probably could be a carbon-based nanomaterial or non-carbon-based nanomaterial. So, and then after that, we need some kind of the binding molecules. It could be enzyme, antibody, or the single-stranded DNA that could recognize the protein target that we will like to detect. And at the end, after the complex form between the antibody, for example, and also the antigen here from the virus target, we could detect the complex formation by, of course, because it's electrochemical, we could detect it by some parameter like a current, potential difference, and impedance. And here I want to focus about the electrochemical impedance spectroscopy, because among the others method here, electron impedance spectroscopy offer more sensitive method for detecting virus. This is the simple illustration about the AES itself. So basically, it's almost similar like what the others method for detecting the infectious virus. Here, once the complex between the antibody here or the biomolecule recognition moiety already formed with the virus target, then we could detect the response. Of course, it's quantitative response here. Once the higher amount of the protein bind with the antibody, for example, here, the higher
like a semi-circular response formed in the device of the AES device. So basically, we could detect and also we could quantify how many virus bind into the device. This is the concept of the AES measurement for virus detection. So here in Park's laboratory, they are quite well known and also expert for fabricating some of the AES-based device for detecting of the virus infectious disease like a norovirus here, and also mosquito-borne virus like chikungunya and also dengue, and also hepatitis virus here in Nature Communication 2-3 years ago, and also even cancer cell. So basically, here I'm coming to the Park Sensor Laboratory the beginning of this year, April 2022, and my task is for fabricating this AES biosensor for virus detection. For fabricating the biosensor for AES and also other electrochemical biosensor, the nanowire or conducting polymer is very important because they will deliver the electron for recognizing the response once the virus bind to the antibody. And here in Park Sensor Laboratory, the polyaniline is very useful, that has been applied for the several biosensor platform. And however, the limitation of the polyaniline here is the emeraldine salt is only conducting state of the polyaniline, which can be obtained in acidic condition, which is almost impossible if we want to construct the antibody-based biosensor in this quite slightly acidic condition. But we could modify the polyaniline and also improve the conductivity of the polyaniline by depositing like a gold nanoparticle or quantum dot or another moiety that could improve the conductivity of this polymer. Here, my task during my postdoc is for fabricating the simple stable polyaniline-based electrochemical impedance for norovirus detection. Is quite famous and also infectious diseases in Japan, especially for elderly. And this is the scheme illustration, the scheme how I construct the electrode for the biosensor norovirus detection. So basically, this is the electrode itself. This is kind of the reusable electrode. We call it gold electrode. This is the gold electrode before we depositing before we modifying with antibody. Here is quite like gold, very fine, and also have a mirror-like character. And then after we have the gold electrode, we depositing with polyaniline here, the green one, and then it's the electropolymerization of the polyaniline, and then we are depositing the kind of nanocomposite between the polyaniline and also gold nanoparticle. The character of the polyaniline, it has a secondary amine here, NH, that could be later on we could deposit or we could modify the antibody here by activating the carboxylic acid group in the antibody by EDCNHS. Then we are blocking the empty space here to minimize the unspecific interaction. Then we are measuring the AES response by this AES machine here. And yeah, in total, this is the kind of the illustration how long it takes for the detection. Then this the first step. We have to prepare the polyaniline gold nanoparticle nanocomposite. Is kind of the simple mixing between the polyaniline and also the gold nanoparticle. Here I'm using several size of nanoparticle, 5, 20, 40, and 80 nanometer. And this is the time result here. The black dark color is belongs to the gold nanoparticle, and yeah, the gold nanoparticle size is less than 10 nanometer here that I'm using. Then this is the kind of a typical Nyquist plot that we get when we are detecting the virus. This is usually once electrode will be scanned 2 times. Once before the virus will incubated. See this kind of the small peak here, and then while there is a virus added into the electrode, the curve will be greatly increased. The radius of the semicircle Nyquist plot will be increased. So this is basically the concept of the AES detection. What I want to show here in the next slide here is the how gold nanoparticle improve the conductivity. Here, the higher radius of the circle here, indicating that the impedance of or the resistance is very high, while here the two graph here, the upper graph here is only polyaniline without any gold nanoparticle, while here the below small one here is the polyaniline and gold nanoparticle nanocomposite, which has much significantly a small radius of the impedance here, indicating that gold nanoparticle is indeed very important for improving the conductivity of the sensor. Then I'm trying to make a kind of a calibration curve and then checking how if we change the gold nanoparticle size in response to the virus detection. And as you can see here, I'm also checking the nanocomposite without any gold nanoparticle, and there is just stop for a while. Okay. May I continue? Yeah. Okay. So yeah, this is the trend of the calibration curve when we are increasing the concentration of the virus and evaluate the dose-dependent response from the AIS. And this is the regression curve. And then what we could see here is 5 nanometer give much lower limit of detection compared to the others nanocomposite. Here, after getting the good result from the previous slide, I'm checking the platform that I have been fabricated by detecting the norovirus, and I'm immersing the nanovirus sample in the human serum, and this is what we got. Is there is kind of a very strong dose-dependent response. If we are increasing the virus concentration, the response also increase. While interestingly, if we change into the dengue virus, there's no interaction, no significant interaction, that's indicating that very high specificity of the sensor. Then I'm also interested to check the platform, the same platform for detecting the dengue virus, not only norovirus. And again, there's kind of the dose-dependent response. While when we are using the platform for detecting norovirus, the very low response was observed, indicating again the specificity of the response. So next, I would like to changing the platform from gold electrode, which is a reusable but quite big platform, to the ones which is disposable but quite handy, and also they're disposable and also portable, using and using this portable ASMS instrument. From the preliminary result, I'm trying to fabricate the electrode for detecting the dengue virus, and it is from our preliminary result. Then yeah, it's quite nice still from the single experiment that could be improved later on. Then this is the feature direction that I want to improve in the future. It's basically in the conventional biosensor, we are still need antibody for detecting the kind of the target virus. But here there is so many research that currently have been performed by constructing molecularly embedded polymer-based biosensor. So it could be some people call it kind of the plastic antibody. So basically, we are using template here. If we want to detect dengue virus, we are using the dengue virus here. We don't need any antibody here, and then the dengue virus will be polymerized with another homonomer, and then it will be like a kind of the hollow vicinity that will be exactly fit with the target molecule. And then we could remove this one and then remove the template that we use for forming this kind of vicinity, and then we could using this platform for detecting the virus. So basically no need antibody for the detection. Then yeah, for the conclusion, sensitive, stable, and universal biosensor platform is important for dealing with the spread of infectious disease, what we are facing during the last pandemic for COVID-19. And then, of course, the existing collaboration between the current Shizuoka University and BRIN improve our national research capacity to develop biosensor platform for detection in infectious disease. And I just want to say thanks to the Prof. Sensei for allowing me to join his laboratory, and also BRIN to allow me to go postdoctoral in Shizuoka University. Thank you very much.
Thank you very much, Dr. Rizal. Indeed, it is very interesting and stimulating many things in our mind. But since we still have another presenter, so let's move to the second one. So for you who have several questions, please keep in your mind and asking later on. So our second presenter will be Indra Memdi Khois, Master of Agriculture, is actually PhD candidate, hopefully soon will be a doctor. And he will present a nano catalyst and its application for virus detection. So Indra, a PhD candidate, the time is yours.
Yeah, thank you for the friendly introductions from Dr. Thawadi. I'm very is it audible? Is it audible? Okay, so my name is Indra Memdi Khois, and I ask permission to share my slides. Okay, is it? Okay, hi. So good morning, everyone, and thank you for the opportunity for me that I can present my works and interest in this mini symposiums together with BRINs. I mean the great institutions, research institution in Indonesia, and with all the fellow researchers. Today I would like to present my interest. Today I would like to introduce my research interest in this limited times. So let me use pointer. So my name is Indra Memdi Khois. Again, I have to introduce myself. I was a PhD student, which I will get my PhD titles next month, I hope. I'm from the Department of Bioscience, a graduate school of science and technology from Shizuoka University. I have my master degree in the same university, and my bachelor degree from Surya University from Tangerang. And my big title is Nano Catalyst and its application for fire detections. So there's three key points here. First is Nano Catalyst, then virus and detections. So each of them I will introduce in the overview. First is what is the viruses and as infectious disease agents. I think we all know about this because of the pandemic of SARS-CoV-2. And the second one, I will a bit introduce about immunoassays as a tools for detection of virus. I think it's also very common and very general for everyone because of the commercialization of the antigen kits from BRIN as well. And the third one is about nanocatalysts, which in here specifically is about nano enzymes. Then nano enzyme in immunoassay applications. Then how a heterostructure nanomaterials as an applied material science is used as an enhanced nanoenzymes and its applications in the immunoassay. Let's proceed. The first one I would like to introduce. So why is virus is the main target in the infectious disease agents, especially in the virus causing disease. So virus, there's two big targets or big factors in here is the antigen or nucleic acids. Of course, there's a lot of biomarker out there, but we will now narrow down to an antigen and nucleic acids detections. And I'm choosing an antigen as the target of interest in the immunoassay because of non-pretreatment required to release or to get the nucleic acid inside the viruses. So antigen is the interesting target for a simple and rapid test detections. Then the final target is the fabrication of on-site biosensor, which should include a rapid, reliable, and cost-effective features in side the biosensor fabrications. So the principle of the fabrications generally is based on the immunoassay. It means that an assay based on immunology body in here is based on the antibody, we can say, which in the ELISA and enzyme-linked immunoassay solvents, the most common immunoassay that was available in the market and in the stage of development, is using a peroxidase. Is an enzyme that was will have an activity that decomposes S2O2 and oxidize a substrate that shows a color. However, as a generator of in the immunoassay, peroxidase, or most commonly horseradish peroxidase, HRP, have a difficulty in the productions and purification. Recently was highlighted, although previously they highlighted on the instability in the harsh environment due to its proteins family. I mean, as an enzyme is of proteins, then it was quite unstable in a harsh environment. Hence, we promote that if we can enhance or if we can change the enzyme into a nano enzyme. So what is actually nano enzymes? Actually, it's coming from a nanomaterials that exhibit an enzymatic reactions or enzymatic activity. So from peroxidase, we propose a gonal particles, a nanomaterials with a nanoscale size that have a similar activity that oxidize a substrate that was colorless initially into producing a color as an oxide form, which with the presence of a hydrogen peroxide or a peroxide species, which shows a tunable activity. This is more important one: tunable activity, resistant, and a high surface specific area. So in the immunoassay, the concept is similar as in the common ELISA, in which that we prepare first with fabricating the platforms with the capture antibody and etc. Addition of the virus to make a bonding with conjugation between the capture antibody with the viruses, and with the nanomaterial that we introduce, or we call it nano enzyme here, it will show a color change due to the presence of this sandwich assay. But however, in the story that we want to functionalize the nanoenzyme for an immunoassay, because the nanoenzyme is just a nanomaterials, we need to conjugate it with an antibody for it to be applicable in a specific immunoassay against our targets. Hence, we need to function with antibody, which we prove by ELISA that we can have antibody on the gonal particles, but not in the one that we didn't introduced. Similar with another ones that we use anti-HIV and anti-norovirus. However, we found that in the previous study that conjugating or functionalization of nanoenzymes pacified or passive patients of the surface of these nanoenzymes, causing a decrease of activity in its application as a nanoenzymes. Hence, we introduced two methods in our research group, using a heterostructure nanomaterials as enhanced nanoenzymes. So what is heterostructure? It means that a nanomaterials which two kind of components, which we call heterostructure or heterocomposite. In the first one, is published in the BNB 2019, combining gonal particles and silver nanoparticles as the coating on the surface of the gold. After the conjugation, we call it silver shell deposition on gold core, which shows a high catalytic activity. And the second one, I will introduce also our recent works in a heterostructure using a platinum nanoparticles and the nano cage using metal oxides nano cage. So the first one, short introduction of the application in an enzyme using the heterostructure using gold and silver. We can see that in here is gold initially, then we can coated the silver on the surface of the gold, showing a double difference structure in the TM, and using the absorbance as a visible light, we can see different absorbance clearly showing the coating on our surface. And we successfully detect norovirus and hepatitis E virus as a monitoring functions based on the RT-PCR functions on the monkey. And we also compare the black dots here, compare with a commercial ELISA, or actually a conventional ELISA, showing higher linear calibrations on our sites using our enhanced Nano Enzymes immunoassay. Proceed. We also would like to introduce the second heterostructure. Similarly, with after the immuno sandwich formation with our heterostructure nano enzymes, it will exhibit a color change when we introduce our substrates, showing the detection of the virus based on the color. Beautifully, on the TM analysis, we can see the oxidize of the nano cage, showing a porous, semi-porous, or we call it pseudo-porous of the nano cage with integrated platinum within the structure. The XRD shows some platinums difference. Of course, more beautiful, we can see the elemental analysis. The blue side here is platinums within the structure of our nano cages. And here also we can see that the important thing is the B part. We introduce a not novel, a derivative factor to examine the nano enzyme in the immunoassay based on the derivative of the kinetic parameter of non-enzymatic reactions, together with some sigmoids analysis, sigmoids curve analysis, which is usually used in the drugs, which is, we call it IC50. In here, we call it NZ50, nano enzymes half reactions. Then we can indicate that the protein nanoparticles within the nano cage is much stronger, or require small amount to exhibit a high activity compared to the normal nano cage without nano enzymes, showing the heterostructure actually improving, showing the concept of nano cage and nano enzyme together. Further, and lastly, the most important thing, we can detect norovirus. We can directly go to the C, that's in the clinical norovirus that we isolated from the human. We can extremely compare our results to the commercial kit from the local company in Japan, that it can show higher linear, wider linear calibrations with higher sensitivity compared to the ELISA kit on the same concept of immunoassays, but using different enzyme and nano enzymes. Selectivity on the B side, showing only norovirus detections, is only because of the presence of norovirus, it will change color, not in other proteins or viruses. And the D1, we can both detect SARS-CoV-2 in case of spike protein and N proteins using specific antibody. So by changing the antibody, we can actually detect specific virus using our nano enzymes. So in the conclusions, I hope I didn't speak too fast, or the conclusion is that we want to detect initially plasmonic chromatic is used as the what's double sound? Used in the commercial kit. Then it go with the super enhanced, published or commercialized by Fujifilm company in Japan. Then we think that it can be go further as nano enzymes for an integrations available platform of immunoassay with nano enzyme concepts. This is our nanobio groups in the Park Labs University. I thank you for the research fellowship from GSPS and the collaborator that provides the viruses and PLP for our detections. Other works in our nanobio groups, some gold positive charge nanoparticle and nano enzymes, supported gold nanoparticle nanoenzymes in a 2D structure, some encapsulation concept with the gold nanoparticles, and some metal oxide nanoenzymes. The last one is electro nanocatalyst for fire detections. That's all. Thank you for your attention.
Thank you, Dr. Candidate Indra. So, ladies and gentlemen, actually, in the presentation is very promising and potential to be fabricated. So hopefully in the next future, we will have another product which is very sensitive to detect virus infection. Ladies and gentlemen, now we came up with the third presenter. Here's Dr. Dodi Irawan Satyo Utama, belong to BRIN organization, and he's graduated from Shizuoka University, and he will present his talk regarding humoral immune response induced with monovalent and tetravalent dengue virus-like particles produced in silkworm. So, Dr. Dodi, please, time is yours.
Thank you.
protein itself for the envelope particle.
Okay. Sorry, I will. Should I restart? Continue. Continue. Okay. Sorry, I forgot to unmute. So really sorry about this. So I will continue about the virus particle itself. So it's a self-assembly and also like in the viral genome. Because, that's why it's become non-infectious compared to the real virus and have a high stability because it's like the real virus itself and can be composed, can be polyvalent structure and have a size in the range of 20 until 100 nanometers. And this can be easily to modify and have a homogeneous in the display of the epitope itself. So in here, to produce the virus-like particle, I use the BMNPV or Bombix mori polyhedrovirus for the silkworm expression system. In here, why I use the silkworm expression system, because this is stable in the production of the protein, and it has a post-transitional modification. Silkworm itself can produce a high level of the secreted protein, and it's easier to maintain, doesn't need aseptic condition, and because this is a very sustainable protein expression system, and it's also environmental friendly. That's why I choose this Bombix mori to produce the PLP. So for my research outline for the monovalent and the tetravalent, so I start from the monovalent, which is I construct the cPRMA and PRMA. This is the I use the synthetic DNA, which is this is contain the C capsid, premembran, and envelope, and also the premembran and envelope of the dengue, and express this to, using the silkworm expression system and purified using the affinity chromatography, and did the biological functional assay for the dengue LP step form using this construct. And then after a biological functional assay, I continue to do animal experiment to see this BLP can induce the humoral response in the mice. So while for the tetravalent, almost similar the flow of the research, but in the to do the expression, I did the co-expression for the construct cPRME and PRME, and this is I will explain later how to do the co-expression. Meanwhile, for the other flow is almost similar until for the humoral response for the this dengue VLP in the mice. So I will continue with the monovalent first. So in here, I construct the genes of each serotype of the dengue, which is contain, I already mentioned, it's contain the capsid, premembran, and envelope. And in here, at first I produce the plasmid recombinant, each of the serotype, and then produce the backmid recombinant. And from with the backmid, with this backmid, each of the backmid is injected to the silkworm, and the silkworm will produce the monovalent dengue VLPs. So this is for the expression itself. In the red arrow, it's shown that the protein is expressed. First, I tried to express it in the larva, and it's expressed in the larva and can secrete it to the hemolymph. And the other is I also did it in the pupa, and it also can be expressed in the pupa for both of the construct, and also in the BF5 cells, which is this is the other type of the expression system included in the silkworm expression system. BF5 cells is the cell line of the Bombix mori itself, and it can also express my protein. So both of the construct for the CPRMA and PRMA can be expressed in the silkworm expression system. For those one is for the dengue 2, while for the other dengue 1, 3, and 4, I only focus in the expression in the larva, which is in this red arrow. It's shown that the protein is expressed and secreted to the hemolymph. And for additional, I also express the E subunit for all serotype. This is for the ITC. This is for the heparin binding assay. So yes, from this we can see that all of the protein can be expressed. So after that, using the affinity chromatography, flag tag, I tried to purify the both of the construct of the protein. And in here, we can see the band in the red arrow that all of the serotype can be purified using the using the affinity chromatography, including the subunit. So means that these all of the protein CPRMA and PRMAs monovalent can be purified by the affinity chromatography. While the dengue 3, because the dengue 3 for the tag is kind of different, I use the PA tag. It also can be purified using the PA tag affinity chromatography. And to see if the construct that I use, it can produce or form the PLP, we need to do the transmission electron microscopy to see the form of the PLP itself, and to check if this PLP can display the epitope that we want. So composed by the epitope or the envelope of the dengue itself. So from here, I use the first antibody, which is this is in here. I use the polyclonal antibody of the dengue, envelope of the dengue, and the second antibody for the HRP. And in here, the HRP is conjugate with the gold nanoparticle. So this can show the surface of the VLP is contain the envelope of dengue or not. So from the result for each the serotype in here, for each for each the construct, we can see there is a small dot, black dot on this structure. So this structure. So this is the gold, conjugated gold nanoparticle, which is this bind to the first antibody. The first antibody bind to the envelope on the surface of the dengue, and this is we can see that this my PLP can form my construct can form the PLP, and this PLP have the envelope on the outer side of the PLP itself. So to continue, if this VLP contain the ED3, because the ED3 or envelope domain 3 is the infectious epitope region in the dengue. So we need to do the heparin binding assay for this, because the heparin can bind to the ED3 site of the dengue. So from here, I use the avidin coated microplates and bind with the biotin heparin, and put my antigen and detect with my first antibody, which is the anti-E, and also the SRP conjugated antibody. And here we can see from the concentration of the protein, and it can bind with the heparin until 20 microgram per milliliter. This is also this is also content for all the serotype. So from the heparin binding assay, we can see that the VLP is contain the ED3. To make sure this ED3 can bind perfectly to the heparin receptor in in the cell. We need to do the ITC to see the affinity binding between the heparin receptor with the ED3. And in here, we can see there is a sigmoidal graphic, which is this is indicate that the binding is can be count, and it's in the good term of binding. So we can calculate and the KD value for this, and for the KD value for the monovalent, I can get in the range of the nanomolar for this, and this range is in the range of the good binding affinity between the heparin and also the ED3. So in here, from the CPRME and PRME binding between the heparin and also the ED3. So we can see the ED3 is present on the surface and the monovalent of the PLP. And for the value, the KD value for the CPRME is lower than the PRME, which is CPRME have a higher affinity to the heparin compared to the PRME. And we compare also for the subunit only for the E, which is this is produced by the silkworm. We compare with the E from the dengue to produced by the cell of the COS by COS7, and the value is can be comparable from the protein of envelope dengue 2 to the envelope dengue 2, which is produced in the mammalian cell. And to see if this VLP have a real have a similar epitope display with the natural dengue virus, we challenge this. We challenge this with the mixed sera dengue patient or human human patient. So in here we can see the binding between the human between the patient sera with the VLP is higher compared with the with the control of anti envelope. And in here we can see that the this VLP have a similar epitope with the natural dengue virus. To see if this VLP can induce the humoral response in the mice, so we inject this. We do the animal experiments for this in the mice. So we inject the this VLP in the biopsy and immunize it for 28 days with the 2 booster. And from the blood that we isolate, we can see that in here the graphic in the graphic that the antisera from the mice it can be bind with their own antigen. So and in here we can see that the CPRME have a better response compared with the compared to the PRME. And for others is we also use the adjuvant to see if this adjuvant can help the response better compared with without the adjuvant. And we can see in this that the with the adjuvant have a better response compared with without the adjuvant. So for the tetravalent vaccine candidate, from the structural gene then, that I already construct for the monovalent, same I use to produce to make the plasmid recombinant and produce the backbone recombinant. But in here I mix all the backbone recombinant by ratio 1:1:1 each. So in here these 4 directly injected to the silkworm itself, and then we assume that this will produce the tetravalent dengue VLP with the variation of the backmeat. For first, we need to check the expression itself, and in here, by detecting with the anti-E polyclonal antibody, we can see there is a band, expression band in the hemolymph, and we also need try to check if this construct, the CPRME and PRME co-expression, have all of the epitope or the variation of the envelope from the each serotype. So in here we can see that each serotype, its construct is detected by the antibody from each serotype, and we can see there is a expression in here. We can see there is a band in here. So this is we assume that this expression can be confirmed and have a variation of the envelope. And we need to purify this expressed protein. And in here, I use the magnetic flag tag purification, which is will save time and also have a better amount of the purified protein compared to the conventional affinity chromatography. So from here, we can see there is a band indicated by the red arrow, which is detected by the polyclonal antibody from the purified protein from its construct. Again, to confirm the variation of the envelope itself for all serotype, I detected using this different serotype of monoclonal antibody, and it can be confirmed. So this means that its monoclonal antibody can confirm the display of its envelope in this both of the construct of this tetravalent VLP. Again, to see this VLP construct can form the VLP, I use the immunotherm with the same way with the monovalent. But in here, I'm not only detect using the polyclonal antibody, but also I confirm with the monoclonal antibody. And in here, we can see there is a little black dot in the attaching to this circular spherical structure for both of the constructs, CPRMA and PRMA. And this also confirmed by its serotype monoclonal antibody, which is its monoclonal antibody. In here, we can see the bind of the gold nanoparticle to the surface of the VLP. And in here, that this construct capable to generating the VLP and also can display for the variation of the serotype. Same to see the ED3 on the surface of the VLP itself. I need to do the heparin binding assay, and from here we can see same pattern, which is the binding of the heparin to the surface of the VLP, and also similar to the ITC result itself. And from the ITC result, we can see that the value if we compare with the monovalent, it's lower than the monovalent, which is this also confirm that the ED3 is present on the surface of the tetravalent VLPs, and the CPRMA tetravalent have a better higher affinity compared with the PRME. And same, we also try to detect this, to bind this with the Mixera dengue patient. And in here we can see compared to the positive control, that the patients have a higher response compared to the only with the anti-envelope polyclonal antibody. So means that this VLP, tetravalent VLP, have a similar epitope to the natural dengue virus. So again, to check the humoral response from this VLP, we need to inject to the mice and produce the antibody, and from the antibody produced by mice, we detect with the antigen, and this from here we can see the CPRMA compared with the PRMA have a higher response, and with the adjuvant also can increase the response for the humoral in the mice. So for the conclusion, for all the monovalent and tetravalent CPRMA and PRMA, it can be formed, and this VLP have a proper structure with the ED3 present on its surface, and this VLP shared a similar epitope with the natural dengue virus, and the tetravalent dengue VLP elicits more immune response than the monovalent. And this, I would like to send my special gratitude to my Professor Eno Park, and also Dr. Sabar for their guidance during my study, and also Dr. Vidya Sata from University of Indonesia, and Dr. Etik Mardiati, Mr. Ali, Nur Ali, Mr. Julham, and also the member of the Laboratory of Biotechnology in Suzuka University, and also the members of the diagnostic kit lab in the BRIN. Okay, I think that's all for my presentation. Thank you very much for your attention.
Thank you so much, Dr. Dodi. So many things, so many data have been presented. So now we end up with the question and answer. I think we're running out of time, but if you have any question, then please raise hand and directly to the speakers. Okay. Yes, please say from Mr. Ivan Mamba Ulmunir. If you would like to open mic and speak directly, maybe better. I just already know. Okay, please. Okay, please. Miss Kaidat Bihandayani. Miss Kaida.
Oh, yeah, thank you very much for the opportunity. I'm sorry for okay, so I have a question to Dr. Dodi. So for the characterization, what kind of methodology you explain? You explain to characterize your monoclonal. Thank you very much. I'm sorry, you are still mute.
Sorry, it's happened again. So thank you for the question, Mrs. Farida. So for the monovalent and also tetravalent, actually I did the same characterization, only with the different antibody for the because I need to check the the display of the envelope for the each dengue in the tetravalent, but basically same. So for the characterization itself, beside the SDS page and Western blot, so I did also the TEM and also immunoTEM to see the forming of the VLP and also the composition of the VLP itself, which is in the outer side, it will be display the envelope. So to detect that, we need to detect it using the immunoTEM, which is almost similar with the Western blot and also the ELISA, but different is we can see the binding between the gold nanoparticle with the surface of the protein if the envelope is displayed. It also can be done in the tetravalent also. And the other thing is I did the heparin binding assay, which is can confirm the ED3, the envelope domain 3 on the surface of the dengue cell, because this ED3 will bind to the surface of the cells. And the other thing is for the humoral, so I did the animal experiment in mice to see the humoral response for both of the construct and also both of the kind of the VLP.
Yeah, thank you very much. Good luck for your study.
Okay, thank you very much.
Okay, thank you, Ms. Faida. Dr. Dodi, this is a question in here. It's from Ivan Mamba Olmone. For Dr. Dodi, I'm just curious, why did you use silkworm as your organism model to conduct your research? Silkworm is an unusual organism model, as I know for gene expression experiment. So please respond it, Dr. Dodi.
Thank you very much, Mr. Ivan, for the question. Why I use the silkworm itself, because the silkworm can have a ability to produce the protein similar to the mammalian itself, and it's easier to maintain because you don't need any aseptic condition, and to take care of the silkworm, it's quite fast to produce the protein itself. And you don't need many amount of the, like if you produce using the mammalian cells, you need much amount for the cell for the expression, while silkworm you only need several silkworm to have a same concentration of protein that you can get compared with the mammalian cell. And the other thing is the silkworm is sustainable, so and it's safe for the environment. I think that's what I can answer.
Okay, thank you, Dr. Dodi, for the response. So I think if there is some other question, please raise hand or just write it down in the box. Okay, so this is because we are running out of time. So just another question. I'm very sorry for this. Siapa nih? Okay, Bu Winnie. Yeah. This is from Bu Winnie. I would like to ask Dr. Dodi, how about antibody neutralization? Did you also examine it? Yeah, please, Dr. Dodi.
Okay, thank you for the question, Dr. Winnie. Actually, yes, this is for the neutralization assay. It's ongoing. But some of data I already got, and it's also already presented in my defense, but I cannot put in here because all of the experiments is not yet finished. So I would like to finish this as fast as I can for the neutralization assay during my time in BRIN here now. So yes, it's in progress. Thank you.
Thank you, Dr. Dodi. I think there is some still question, especially for Dr. Rizal and also for Mr. Inda. But since we are running out of time, so I'm very sorry to close this session. So please give a big applause for our presenters.
Thank you so much.
Thank you so much, and I would like to turn it to Dr. Eka. Thank you.
Seems like we all heating up already in this session, on the second session. So the highlight is actually we go from the basic to the applications, and we have gone through all most recent updates in regard with what's it, biosensors and diagnostic tools. We are going to move on very much downstream application of another application that is more into antibody monoclonal, which will be facilitated by Dr. Astuti Nurhasana. Pardon me. So, floor is yours.
Okay, thank you very much, Dr. Ika Nurlaila. Good afternoon, ladies and gentlemen. It's an honor for me to lead this session, who is an all-ladies session. Since we are actually almost half an hour behind the schedule, I would just like to introduce the three speakers that are going to deliver their speech today. The first one is Ms. Tika Widayanti, from Center for Vaccine and Drug Research, and then the second will be Ms. Asri Sulfianti, and the third will be Ms. Febi Nurdianingsih, also from the Center for Vaccine and Drug Research. The first one, Ms. Tika Widayanti, has got a master's degree from the Faculty of Medicine, University of Indonesia, and at the moment she's going to deliver her presentation in the development of monoclonal antibody anti-spike SARS-CoV-2. Please, Ms. Tika Widayanti.
Thank you, Buti, for your kind introduction. Good morning, ladies and gentlemen. My name is Tika Widayanti. I would like to present our recent research about development of monoclonal antibody anti-spike SARS-CoV-2. SARS-CoV-2 virus is caused coronavirus disease that spread globally and became the major health problem since today, with the death tolls worldwide more than 6 million people. On the left, it's the genome of SARS-CoV-2, which contains 4 structural proteins, including spike protein. The spike protein plays the key role in initiating viral integration into the host cells. These important roles made the spike protein as a target for drugs and detection. So what is a monoclonal antibodies and why we should develop this molecule? Basically, monoclonal antibodies or MABs is a monospecific antibody that selectively only bind to one epitope. The advantages of MABs are MABs can be produced in laboratory, can be engineered, modified, and its properties mimicking natural immune response. MABs has been used widely as reagents in research field for diagnostics materials such as lateral flow assay or immunodiagnostics, and therapeutic applications for cancers, infectious disease, and so on. The picture on the right is one of the method generate MIBs. The fusion between B cells from mice, splenocytes, and tumor myeloma cells resulted in hybrid cells that can produce MABs. Therefore, in this study, we generate a monoclonal antibody for spike protein from SARS-CoV-2 that have crucial role in penetrating host cells and initiating infection. In order to generate hybridoma MABs producing cells, the first is to clone and express the spike RBD protein in E. coli. Hereafter, the recombinant protein was injected to biopsy mice with a 2 weeks interval. Hybridomas were obtained by the fusion between myelomas and B cells from immunized mice. After screening for potential hybridomas, MIBs were produced by injecting hybridoma cells back to the mice, or another method is hybridoma cultivation in cell culture medium. The MABs purification is performed by using affinity chromatography. For protein analysis, MABs were tested by indirect ELISA and Western blot imaging. So here's the result. Hybridoma cells generated by cell fusion between spleen cell from immunized mice and myeloma cells, as shown in left figure. The screening of potential MABs were performed with indirect ELISA. 5 wells with the highest absorbances were selected for further step to obtain single hybridoma clone by limiting dilution. Monoclonal antibodies were tested against recombinant spike protein and whole inactivated virus. In this study, we use CoronaVac or Sinovac vaccine as antigen source. The hybridoma selection with indirect ELISA showed 17 clones that potentially produce anti-spike antibodies. However, 4 out of 17 MAB clones were consistently shown positive result by ELISA analysis against inactivated COVID-19 vaccine and recombinant spike protein. In order to get purified MABs, purification process is one of crucial steps in MAB production. Purification is aimed to eliminate lipid particles, cell debris, host cell proteins, and other contaminants that can affect antibody activities. MAB purification was performed using affinity chromatography with protein G resins with following conditions. Elution stage were done using glycine chloride buffer with extreme low pH. MAB characterization was performed by indirect ELISA and Western blotting. From the left picture, it shows SDS page visualization of certain fraction from purification process. There are 2 protein bands in size about 55 and 28 kilodalton that indicates the heavy and light chain of antibody structure. In the middle picture shows ELISA result from purified MABs fraction 19 and 20 against a whole inactivated virus. Seen that absorbance from purified MAB significantly high compared to negative control. Fraction 19 also used in Western blot analysis, which performed using 293 transfected cells with full spike recombinant protein. The result shows one band in size about 30 kilodalton that matched to the size of RBD protein. As I mentioned earlier, monoclonal antibodies can bind to certain targets such as antigens on the surface of cancer cells, viruses or proteins in solutions, or body fluid in detection system. Affinity chromatography using protein G or protein A resins are often used in monoclonal antibody purification process because they are stable and selectively binds to Fc region of immunoglobulins. MAB's anti-spike could recognize RBD recombinant protein that expressed by transfected 293 cells. From this study, we can summarize that monoclonal antibodies which obtained in this study could recognize the RBD recombinant protein and also whole inactivated SARS-CoV-2. These findings make the anti-spike MABs as a potential biological material that could be used in the development of SARS-CoV-2 spike protein detection system, but the further analysis should be conducted. And this is our team. I would like to say my gratitude to Pak Masteria as head of Research Center for Vaccine and Drugs, monoclonal antibodies team, Pak Sabar as the leader, Asri, Febi, Jody, Mbak Ika, Ayu, and Angel at Research Center for Vaccine and Drugs, and thank you for your attention.
Okay, thank you very much, Tika. Now let's move on to the next speaker, Ms. Asri Sulfianti. She also graduated from the University of Indonesia from the Faculty of Medicine. Please, Asri, she will be presenting formulation of gold nanoparticles ablation technique with anti-spike SARS-CoV-2 monoclonal antibody as raw material for COVID-19 detection.
Yep. I guess my voice right now can be heard by everyone. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, Bapak dan Ibu sekalian. Good morning to afternoon for everyone. I am Asri Sulfianti, and today I'd like to present our team research, which title is Formulations of Gold Nanoparticles Ablation Technique with Anti-Spike SARS-CoV-2 Monoclonal Antibody as raw material for COVID-19 detections. The COVID pandemic in Indonesia has prompted Indonesian pharmaceutical companies to develop rapid testing to detect the spread of virus SARS-CoV-2. But both detections with real-time PCR and rapid antigen detections has been used in Indonesia are imported from abroad. Even the real-time PCR is a gold standard for detect the SARS-CoV-2. This method is quite expensive and need a competent people to use it, while rapid test detections is become an alternative to solve this problem. However, mostly rapid test detections in Indonesia and its component are imported and pricey. And to strengthen the structure of domestic industry and reduce dependency on imported products, our government regulations issued the use of domestic material by increasing the domestic content level, which targeted to 40% in 2020 and 50% in 2024. This reality automatically encouraged the Indonesian researcher to do innovations by employing local resources. And in 2020, the Center for Pharmaceutical and Medical Technology and now merged into the Center for Vaccines and Drug Research, BRIN, has succeeded develop the hybridoma cell-produced anti-spike SARS-CoV-2 monoclonal antibodies. This hybridoma was made from fusion between myeloma cells and B cell of mice that induced by RBD antigen. and the lab scale productions in vitro culture has also succeeded yield the monoclonal spike, which can detect the commercial antigen of spike, and also can detect the attenuated SARS-CoV-2 viruses in Sinovac vaccines. And separately, Physical Research Center and now become Photonic Research Center in BRIN has also made a gold nanoparticle by using laser ablation method. In this technique, the destructions of salt golide in water medium under laser radiations can create a colloidal suspensions, and because in this method no additional reagents were been using to synthesize the GNP, so the GNP are obtained are quietly pure. So if these two materials can be used together, it can be available as a raw material for COVID-19 rapid detection kit. Thereby, in our study, our main goal is to formulate the anti-spike immobilizations on the preparations of laser GNP, and also to determine the minimum concentrations of antibodies needed in order to get high antigen capturing activity. However, before we formulate these two components, there are several steps that we need to prepare before. First of all, we do the productions and purifications of anti-spike SARS-CoV-2. Productions of anti-spike was carried out on SF2 clone that grew well in on RPMI with 3% of FBS and serum-free media. The reason why we selected the clone that survived to grow in a minimal serum media content, because it can minimize the other protein contamination during purifications, and based purification with G protein resin resulted that the targeted antibody was only seen on a clone that grew well in RPMI with 3% of FBS, as we can see in the picture that the targeted protein or antibody migrated like to protein band, which migrated at 50 kilodalton for heavy change of antibody and about 28 kilodalton for light change of antibody. Unfortunately, the protein target cannot be seen in the productions of the antibody in SVM or serum-free media contain. The analysis for the antibody that produced in 3% of FBS revealed that the absorbance of the fraction 2 was about 0.7, and this quantification with BCA assay, the protein resulted about 292 microgram per milli protein. So, and then the second step that we need to do was GNP synthesized by laser ablation technique. The destructions of a solid gold with 30,000 shoot in water medium resulted in a gold nanoparticle absorbance about 0.5 with a maximum spectrum at 520 nanometer. As we can see at the curve, the range maximum of the absorbance started from 500 until 540 nanometer, and the characterization with dynamic light scattering or DLS revealed that this GNP has monodisperse with average diameter was about 44 nanometer, and another characterizing with zeta potential showed that the GNP was highly stable because it can because it has a surface surface charge about minus 38.5 microfold, sorry. And another step that we do is how to stabilize our GNP with a stabilizer component like BSA. Additional salt on GNP BSA conjugate, as we see in this picture, no color changing. Capping GNE with a stabilizer component such as BSA indicated that the color of GNP didn't change. Even at the lowest concentrations of BSA at 31.25 microgram per mil, showed that the GNP was stable with a bare minimum of BSA. And theoretically, the GNP aggregations will observe if we measuring it in a wavelength of 580 nanometer, and as we see in the picture, at the beginning, conjugation with BSA increasing the absorbance of GNP, but it leading to the plateau condition, which indicate the saturations of the GNP surface with absorbed molecule. So it is indicated why no shifting color on GNP if we use BSA as a stabilizer. However, when we try to conjugate the anti-spike, our anti-spike SARS-CoV-2 with the laser GNP, according to our experiment, no concentrations of antibodies can stabilize the GNP. It is indicated by changing color of GNP from red to blue because of shifting in the surface plasmon response of the GNP, or the SPR rising can be observed by the absorbance at 580 nanometer. Yeah. And as we see in the picture, increasing concentrations of antibodies trigger the decompositions or aggregation, which indicated by the elevations of absorbance, and we see that no concentrations of antibody was able to stabilize this GNP. So what's problem with our anti-spike? As I mentioned before, that when we visualize the purification result, it produces a good characteristic. But if we try to check again our anti-spike with SDS page, that the result is no band targeted seen for heavy or light change. This is probably the reason why anti-spike cannot be found to stabilize the GNP at every concentrations. The loss of antibody, it is because it was too long to be safe at elution buffer that contain glycine at acidic environment. Theoretically, because in acidic environment, a greater percentage of glycine molecules become positively charged. On the other hand, our GNP charge was negative. At the beginning, we presume that it will stabilize the GNP, but however, conjugation of to this material with additional of sodium chloride result in changes in electrical double layer of GNP and shift in the equilibrium between electrostatic repulsions and attractions of particle. This is maybe the reason why we can found a stabilized of GNP by our antibodies. And for conclusion, the productions and purifications of anti-spike SARS-CoV-2 was successfully demonstrated that the antibodies was found as a 2-protein band target at 55 kilodalton for heavy and 25 kilodalton for light change. And the GNP synthesized by laser ablation technique was yielded 44 nanometer GNP that has a charge surface at -38.5 millivolt. However, the conjugations these two components were failed. The antibodies couldn't stabilize the GNP because it was lost during storage in acidic environment, and shifting in the equilibrium between electrostatic repulsion and attractions of particle are the hypothesized reasons why the stabilizations of the GNP couldn't be reached in this research. That's all my presentation today. Thank you very much for your attention. And this is our team, monoclonal antibody research group that led by Dr. Sabar Pambudi, and also special thanks for Dr. Isnaini to provide the GNP, and also from special thanks to Dr. Etik Mardianti and Dr. Rizal for the technical supervision during the research. Thank you.
Okay, thank you very much, Ms. Asri Sulfianti, for the very interesting presentation. Let's move on to our final speaker for today, Ms. Febi Nurdianingsih. She will present an adaptation of the hybridoma cells producing monoclonal antibody in low serum media. Please, Febi.
Check. Could you hear my voice?
Yeah.
Yes. Thank you for the chance, Dr. Titut. Let me introduce myself first. First, I would like to say good afternoon to everyone. I'm the last presenter of today's event. So let me introduce to myself. My name is Febi Nurdianingsih. You can call me Febi, and now I'm working in the team of monoclonal antibody with Dr. Sabar and everyone in the monoclonal antibody team. And today I would like to share about my presentation entitled Adaptation of Hybridoma Cell Producing Monoclonal Antibody in Low Serum Media. So as we know that monoclonal antibody, or MAB, is a biological material that widely used for many applications, such as the development of kit diagnostic, and also used for therapeutic for certain diseases such as cancer, and also immuno-based assay that usually we perform every day in our laboratory. And I believe that previous presentation also has explained about what is a monoclonal antibody. Yes, monoclonal antibody is described as an antibody that produced by B cell that specifically recognize the epitope of foreign protein or antigens. And also there are various platform technology that perform to produce the monoclonal antibody. But in our laboratory, we use a hybridoma technology to produce the monoclonal antibody. And sorry. Yes. In order to produce the large scale of MAB, it's commonly performed using in vitro method by cultivating the hybridoma cells in the culture medium, supplemented with animal-derived serum, such as FBS, inside of the bioreactor or fermenter, which is maintained under optimum condition for the cell to grow. And also, we know that serum is known as the main nutrient source, also growth and proliferating factors for the cell, which is the use of serum in the culture media remains unavoidable. However, the use of serum in a culture media for large-scale production takes a really high cost.
In addition, there are some disadvantages resulted from using of a serum in the culture media because it gives high impurities during the purification process of the MAB itself. And also, serum may has a large variability of content due to batch and also source and environment, which can ultimately lead to the reproducibility problem in the experiment, since the environment may have influence on the serum content. Also, there is the risk of pathogen contamination must be considered too, and we know that the process to obtain this serum from the animal may have the ethical problem. Overall, the use of the serum is not sustainable at all. Then we came out with the idea, is it possible to grow the cell in a minimum serum condition? And so we can produce the large amount of MAB in a minimum cost. But first, we have to adapt the hybridoma that we have in the low serum condition. Also, we need to evaluate the growth and MAB production kinetics from hybridoma that we have in low concentration of serum. So the next we can compare which one of the clones that have high potential to grow in the low serum condition. So we have two clones that adapted and evaluated in this experiment. It is SF2 and RF10 clones, which both clones produce the antibody against the SARS-CoV-2. Generally, the adaptation process was performed gradually start from 10% of FBS supplementation as the initial culture condition, and then cells were cultivated in a well plate with the density 100,000 per milliliter under the optimum condition with a 37 degrees Celsius and also with a 5% of carbon dioxide supply to make the cell is grow well. And then after 8 days adaptation in certain stage or around 3 times cells passage, the hybridoma was passaged into the culture condition with a lower FBS concentration. And in the every stage, we collect the sample of the culture media to measure the MAB production by ELISA analysis. We measure the optical density in 450 nanometer. Also, we upscale the cells number to make the cryostock of each stage. And that, sorry. So, here okay, sorry. So the next is after the hybridoma were well adapted in the media supplemented with the 3% of FBS, with the lowest concentration that we can reach, the cell were cultivated in the T-flash until reach the confluency at least 80%. Then the hybridoma was cultivated in spinner flash with the working volume 70 milliliter with this condition for cell to grow. In this experiment, we use batch culture system since we want to monitor the growth of each clone in low FBS condition for certain period. The growth of cells were monitored for 11 days. Actually, we usually use 5 or until 7 days, but in this case, because we grow the cell in the very low concentration of FBS, so there is a possibility that cell will grow slower than the usual. So we decide to observe it for 11 days. And then we monitor the growth of cell every day.
by counting the number of viable cells, and also we collect the medium culture for antibody measurement by indirect ELISA, using the commercial spike recombinant protein or whole inactivated SARS-CoV-2. So here the data of the production of monoclonal antibody from the clone SF2 in the culture media supplemented with the various FBS concentration. There are 4 states. One is 10%, 8%, 5%, and then 3% is the lowest concentration of FBS that we use here. And for the positive control, we use the commercial antibody